Assalamualaikum. Thank you, sir, for the invitation and for the opportunity to present my work, efficacy of bacteriophage therapy in controlling Salmonella enteritidis infection and reducing environmental contamination. As we all know, we are facing a slow pandemic that is antimicrobial resistance that is rising with uncontrolled use of antibiotics, especially in poultry. We have a uncontrolled and unethical use of antibiotics in poultry. And Salmonella enteritidis is a public health issue that is zoonotic infection can be transferred from animals to humans, and it is causing almost $3 billion losses to the poultry industry worldwide. And bacteriophage, we are looking for the alternatives to the antibiotic, and probiotics and bacteriophages are among the alternatives and the biocontrol to control the pathogenic bacteria. The bacteriophages was isolated from the sewage from the various poultry farms. That work was conducted by my senior, Dr. Alina. They were isolated from sewage sample. There were 17 bacteriophages that was isolated, and among the 17 bacteriophages isolated, 3 was lytic bacteriophages that has much more efficacy against Salmonella enteritidis in vivo, and I did the in vitro trial at Poultry Research Institute, Rawalpindi. For that purpose, we procured the desi chicks from the market and housed them at the Poultry Research Institute, Rawalpindi, and we use Salmonella enteritidis species from our research institute, UAS, Lahore, and then we use the cocktail of SELF1, SELF13, and SELF20. SELF1 belongs to Cyphovirus family, and SELF13 and 20 belong to Myvirus family. And we use the quantitative bacteriology to check the efficacy of the bacteriophages. The experimental design, we divide the birds in 5 groups. First one, the negative control, in which no treatment and no infection was used. The second one was the positive control, in which only infection was administered by oral route, and the dose was 10 to the power 5 CFU per ml. And then the treatment groups, prophylactic group and the 2 treatment groups. In prophylactic group, we administered the bacteriophage at the dose ratio of 10 to the power 7 plague forming units PFUs on day 4, 5, and 6. And then we administered the infection on day 7. While in delayed post-challenge group, we administered the infection on day 4 at the dose rate of 10 to the power 5 CFU per ml. While in 6-hour post-challenge, we administered the treatment, that is the cocktail of 3 bacteriophages on the day 6 of the chicks. Then we collected slaughtered the birds and collected their cecum samples, and then picturate them, and we do the quantitative bacteriology to observe the log reduction of the bacteria. First one is the comparison of the different. We use 3 different routes to administer the bacteriophages. One was the oral gavage, second one was the drinking water, and third one was the aerosol route. This is the positive control group. You can see there is a high load of Salmonella, and these are the different groups. First one is oral gavage, that is on the day 3rd of the post treatment, day 7 and day 14, and you observe that oral gavage route has much better result as compared to the aerosol route and drinking water route. Now we go next is the delayed post-challenge treatment. In this same oral gavage has much better efficacy as compared to the drinking water and aerosol route. And last one is the 6-hour post-challenge. In this we also observe the same, because Salmonella affect the JIT of the bird. So that's why the oral route is much more effective than the aerosol or drinking water route. If we compare all 3 groups in a single slide, we observe that prophylactic group has much better result as compared to the post-challenge treatment, 6 hours post-challenge, and delayed post-challenge group. Conclusion of the my study was that the bacteriophage has an effective result against Salmonella enteritidis, but there is still a problem because bacteria has its own defense system that is CRISPR-Cas system, and the restriction enzymes that degrade the bacteriophages. We have to overcome that thing to launch this product to use the bacteriophages as a biological control to Salmonella or pathogenic bacteria. Thank you.